So the second half of
Chapter 5
chapter
talks,
5, look at the energy
moving
and
into
energy in
a
terms of how it's used in
more
living
complex
things.
discussion.
So there's
of
some
of the stuff we
definitions that you
introduced in
have to know.
chapter
And
4.
so starting
we wanna
one
really
is
take a look
energy
at
is the
things
capacity
like
to do
solid
work.
boundaries.
We can
Including
think about it
the
as the
plasma
ability to
membrane,
create
that's
order
what
from
we'll start
disorder.
off
If you
with,
think about
with
cells as being
chapter
organized
5.
things,
So
obviously it's going to
the
take
cell membrane
a
or
lot of
plasma
cellular
membrane,
energy
there's two
in order
different names
to
for the same
create
thing.
these beautifully
Again,
organized cells.
There
There
are
are
different
different forms of
terms
energy,
used
kinetic, potential, and
synonymously.
chemical energy. And
The
we'll go
membrane
through
is
those
made
and
up
make sure that you're comfortable
what we
with
call a
those
fluid mosaic
various
model.
ideas.
So there's clear things that you can see. We've talked a
so
lot
let me go
about the fact
on
that there's a phospholipid bilayer. But this
and
membrane
talk
is
about
made
what
up of
cells
many
do
other things.
with
It's a
energy.
mosaic of
And
several
so
components.
we can
And
start
wanna
off
look
by
at all the
saying,
components,
thinking a little
what
bit
they're
about
doing
what
there,
you do with energy.
and again,
if
how
you
they
ever
interact
think
to make the
for a
membrane,
moment about why you eat.
characteristics
One of
that it
the
does.
main reasons you
Now,
eat
again, this
is
is Biology
that
101. We're
you
doing
need
a somewhat
energy in order to
simplistic
carry out the
discussion
activities
of this
for what your
cell membrane.
doing.
And there
So
are
if you think
some
about it,
mistakes
eating
in the
is
text. And I'll
tied
point those
to
out as we go
whatever it
along.
is you do. Eating is tied
So
to
let's talk
learning.
about the
Without
phospholipid
eating, you
bilayer.
couldn't have
For
the
the
energy
most part,
available
the
to learn.
phospholipid bilayer
If you exercise, eating is tied
going
to
to make up most of the
exercise.
cell
Without
membrane.
eating, again, that
You
energy in, there
need
is
to
no
know that
energy
there are
out.
polar heads.
So again,
and
when we think
nonpolar
about
tails,
cells
on the
couple
phospholipids,
reactions
and the
in which they
nonpolar
require
hydrophobic tails
energy
stick together.
because
any
reaction that requires energy will be
they're
coupled with
repelled by the forces or
reactions
polar
that
forces
make
of
energy
the watery
available.
insides and outsides
So
of the
again,
cells.
cells use
holds the
energy
membrane
for a
together
variety
kind
of
of like a
things. They
sandwich.
use energy to build molecules.
This membrane
If
is
you have ever seen a
semi-permeable.
little
There are
kid
some
grow
things
in
that
the
can
years
make their
to
way
run
through.
that's chemical
One
work.
of the problems with
Mechanical
this
work could
phospholipid
describe
membrane,
moving
one
and
of the
dividing.
characteristics of this phospholipid membrane
Electrochemical
is
work
because of the
involves
non-polar center,
creating concentration
generally
gradients.
polar molecules
Some
can't make their
definitions.
way
First
through the
definition
phospholipid
is
bilayer.
what is kinetic energy.
Okay,
Kinetic
so
energy is
we
the
say
energy
the
of
phospholipid
movement.
bilayer
Take a
is
moment,
semi-permeable.
take your hand,
Things like
and
oxygen, carbon
wherever it is
dioxide,
right now, raise it
very
up into
small,
the air
very
and put it back
non-polar
down.
molecules
That
can make their
movement
way
was
through.
kinetic energy.
Again,
This potential energy is stored
idea
energy.
that water molecules diffuse or make their
and
way through
this
the
is a
membrane
really key thing.
isn't
We went through
accurate.
all
We've
that trouble
learned,
of learning about
that there are actually
chemical
proteins
molecules
involved in allowing water
I want you to
transport
take just a
through.
moment
So
and think about
the
every time
phospholipid
we saw this
bilayer is
complex
only that
molecule,
small
every time you saw a
non-polar membrane
covalent
make their way
bond,
through.
almost always
Now,
there's
what doesn't
energy
go
stored in
through
the
the
membrane.
membrane
Includes
includes
large
so
potential
molecules.
energy
Large
is stored
molecules
energy,
can't pass
and energy
between
can be
these
stored in chemical
phospholipid
bonds.
heads, as well as water.
We can also have energy available in concentration
ions,
gradients.
they really can't make their way through.
And we know that the breaking of bonds tends
So
to
again, we
release
know that the
energy
major component
and make it
of the cell
available
membrane
to
is
be used.
the
Kind of like
phospholipids, but a secondary component is
chemical
the
bonds are
proteins.
kind of like this guy with the
do a
rubber band
variety
pulled
of
back.
things.
They're
They float
stored
within
energy
the cell membrane.
when the stored energy is
Some are
released
anchored in place
kinetic
with the
energy,
cytoskeleton.
can be used.
They can span
There's a couple definitions you need to
bilayer,
have,
or they can
and
be
again, when you
exposed
see
to
an
one side or
asterisk,
the
that means it's
other.
going to be on the exam.
And many
so
of them
first
are
law of
attached to
thermodynamics is key.
carbohydrates.
It says
attached
energy
to
cannot be
carbohydrates can be
created or
glycoproteins,
destroyed,
or when
so
attached to
we
lipids,
can't
we call
make
them
energy, we just can
lipoproteins.
transfer it from one form to another.
So what are those proteins doing?
And
Again,
then
we talked about
the second
the fact
thing
that
is
some of them are very
energy is
important
always
in,
conserved.
maintaining
This is sort of implied by the
cytoskeleton.
first law or
So
the
they
first
are
statement.
an attachment
That means all
point
energy
for the
entering into a
protein
chemical
of the
reaction must come
cytoskeleton.
out. But it may come out in a different form.
proteins
Okay.
actually are important.
Second law of
in holding
thermodynamics
cells together in tissues.
says that whenever
Transport
a
proteins are
chemical
probably
reaction
the key
occurs,
one
not
that I
all
want you
energy
to
that's
focus on
released
right
is
now.
available to
Transport
do work.
proteins
And some
allow materials to move
entropy
into
is
and outside
lost
of
to
the cell.
disorder or
They can
randomness
actively or
in the system.
passively transport
The
things in.
randomness that we've learned about
There are
so far
receptor
has
and
been
recognition
Brownian motion,
proteins,
and that idea of
which are
kinetic
important
energy or
for
Brownian motion, recognizing where energy is lost
and
at.
also
Whenever
recognizing
energy
cells from
changes
the outside.
form, we're going to lose
And
some
then
of
there
the
are
useful
enzymes,
energy,
sometimes found in
and
membranes as well,
molecular organization
that are important
in
in
terms
become
of
somewhat less
promoting orderly. That
biochemical
lack of
reactions.
orderliness is
We see
called
those enzymes
entropy.
more in the internal membrane.
Entropy is non-useful
So
energy.
when
That's
we start
important.
talking about what moves
Second
into
law of
and out of
thermodynamics
a
is often stated
membrane, we
as
need to talk a little bit about the
entropy
watery
in a system
environment
always increases.
inside and outside of the membrane.
Here's a beautiful
so a cell
sort of
actually is a
description of
combination
energy
two or more
entering
substances.
living
Things
systems.
like,
For what it's worth, we've talked
complex
about the
mixtures.
fact
So you could
that
say all of the
energy
stuff that's
enters
inside of a
most
cell
living
could be
systems
considered
through
a
the carbon
mixture.
cycle. That
As
energy is
opposed
taken
to a
up by
solution,
plants, and
a
that plant
solution
is used
is
in a variety of
something
ways
a solid or a solute mixed
by
in a
those plants
solvent,
as well as by things that eat the
liquid
plants.
that has
Again,
a homogeneous
there's a cycling of
characteristic.
energy, there's
So it all has
cycling
the same
of molecules
characteristics.
throughout living systems.
Again, seawater, honey, gasolines are examples of
And
solutions.
again, we're thankful for the
solution
sun.
has
The
a
sun is the
solvent, which
ultimate
is the
input
liquid
of energy
and a
for all this
solute.
organization.
And we often measure
And a couple
the
more
solute
definitions.
solvent by
Chemical
different
reactions are
scientific
going to
measures.
describe
We use the terms
how
molarity and
energy is
molality,
transferred.
and they're
The
the
things
number
that
of
go
moles
into
or
a chemical
molecules
reaction are called
in a liter
reactants
of
or
solution,
substrates.
We
the
also
things that
look
are
at
produced are
percent
called
solutions, like the
called
percent by
products.
weight or percent by
Every
volume
chemical reaction
in
in the
terms of
cell
the amount
requires
of
an
solute
enzyme or a
in
biological
the solution.
catalyst. When you look at this reaction on the bottom, wouldn't
So
occur if an enzyme
equilibrium
wasn't present.
describes
When these reactions occur,
there's
we
an
break
equal concentration
form chemical bonds, we transfer
substances
energy,
throughout.
we
And
transfer
so a
electrons.
solution by
So you do
definition
need to be
is
reminded
in
that
equilibrium.
reactions that
But
lose
if we put a
electrons,
membrane
we say
between
are
one solution and
oxidized.
another, we
So
can say
Leo,
that there's
lose
isn't
electrons
equilibrium
oxidized.
between these two different
And
states.
if you gain
When
electrons,
we're
you're
in
reduced.
equilibrium,
Things
these
are
are
the
the
most
most
random
random
and most
Leo
stable,
says GER, and they contain
lose electrons
the lowest amount
oxidized,
of potential energy.
gain electrons
Again, if we move
reduced.
things out of equilibrium, that's a place that we can store
There
energy.
is a
and
tendency,
see
again, we
that
tend to
cells
show
take advantage of the
reactions like the previous
can
one as
move
being
things out of
mostly one
equilibrium,
way,
and then
most
allow
reactions actually
the
are not
energy to be released until equilibrium.
are going to
is once
proceed
again
until
achieved.
equilibrium or a balance is
The concentration
achieved.
gradient
And
describes
enzymes can shift
a
the
difference in
equilibrium
concentration
towards the formation
between
of
one area
of
and
one
the
product
next.
over the other.
A good example of that, when you start a
Again,
fire, you'll notice that
let's
there's
start
smoke
talking
right by the
about
fire.
gaining electrons and
I
losing
can't smell the
electrons.
smoke
Generally,
further away,
reactions
but
that
over
lose
time,
electrons,
the concentration
that
of smoke
occurs when
will
you
diffuse
break
out
chemical
away
bonds.
from
So if you lose
the area where it's
electrons,
most
you
concentrated
tend to
to where
break
it's less
chemical bonds.
concentrated. Again,
Again,
there's a
lose
concentration
electrons
gradient
is
where there's more
oxidation.
smoke.
That's
near
where
the
we
fire,
release
less
energy. So
smoke
a reaction that
further away.
releases energy is exergonic. It's catabolic.
Again, molecules tend to
oxidation
diffuse or
occurs.
to move passively
This
from
is
areas where
important.
they're very
This is one of
concentrated to
those.
where they're
Make
less
sure that you write a.
So this is again
a flashcard.
a quick discussion
Again,
of
exergonic
diffusion.
chemical
Diffusion
reactions
is the movement of
release
materials from an
energy.
area of high
They
concentration
tend to be
an area of low
catabolic or
concentration
breaking things
to
down,
concentration.
and they tend to be oxidative.
I really need you to
Endergonic
memorize
reactions are
this-
the
this
opposite.
first bullet
Endergonic
point. You really
reactions
do need to
consume energy, so you
memorize
have to have
that.
an input of energy.
And so you can look at these green dots. If you look at this picture
when
here,
you input energy, you build
the
usually
green dots
larger molecules, so they're
at high
anabolic,
concentration,
and they trap
on this side
electrons or
low
they gain
concentration
electrons.
on the
So
other.
reduction
Purple
occurs.
dots, high
A good
concentration
example
on
of
this side,
a
low concentration on the
endergonic
other.
chemical reaction here is
And you
showing
can say
carbon
that there
dioxide
is a
and water
tendency,
with the
because
help of
molecules
solar
are,
energy and lots of enzymes.
At higher energy, when
produces
they're not in
glucose and
equilibrium, there's a
oxygen
tendency
through
for
the
things to
process
move
of
into
photosynthesis.
equilibrium.
Again, this is
So
a
you can see
good
there's a net
example of an
movement
anabolic
from high
reaction where we're
concentration
taking
to
less
low
complex
concentration of both
molecules
the
and
green
making
and
something
purple
complex.
balls until
The
equilibrium
next thing we
is
need to
reached.
learn about is a little bit about ATP.
Again, this is a
So
passive
again,
process.
ATP
The cell doesn't have to
is
put any energy
how
into this.
cells couple the
Again,
reactions
why
that
do they
release
move?
energy with couples
These things
that
wouldn't move
or
if they were
enzyme
stuck
with reactions
in
that
place, but
require
molecules
energy.
are
We can store
constantly
energy
moving.
in ATP,
And so
and
there's
then
a
make it
natural
available
kinetic energy
whenever energy needs to be released.
Molecules
and
are
then
always
we can,
moving
again, input more energy
by a
into
type of random
taking
motion that we
ADP
call
and P to make
Brownian
ATP.
motion.
We keep cycling.
They tend to
as we
move
need
towards
energy
a
or
more
as energy
random
becomes
state.
available.
And the
Quick
speed that they
picture
move is
of
called the rate of
ATP,
diffusion.
and you'll see a nucleotide.
So the things that affect the
adenine,
rate of diffusion
the sugar is
include
ribose,
temperature, the
and the
steepness of the
phosphate. You
gradient,
can see
and the
that there's
molecular size.
3 phosphates,
Okay.
2 high energy
So
phosphatidyester
diffusion is very,
bonds.
very important in terms of movement of molecules into and out of cells.
Again, this is
I
a
always
repeat of some of
remind
the
people to take a deep breath,
other
breathe in some
slides that I've
oxygen,
had just
and think about the
previously.
fact that the reason
Last
that you
bullet
breathe in
point,
is you
though,
want oxygen
I want you to think
diffuse into
about,
your cells.
and that is whenever
again,
we have electrons,
that
electrons
idea
are
of
used to make
oxygen making
bonds.
its way into your body,
Wherever
very
there are
important to
bonds,
note
we
in
have
your
energy
life.
stored.
And again, it's
Whenever
a
we break
one of the
bonds,
simplest
energy is
examples of
released.
diffusion.
So
in a
if we're
living
building
thing.
bonds in an anabolic process,
Let's
we're
go on and talk
be
about other
storing
types of
energy,
diffusion or other
and if
types
we're
of
breaking
transport.
bonds in a
And,
catabolic
the next type
process,
of transport I want to talk about is facilitated diffusion.
And again,
Now,
here's a
this
picture
is
of
simply
catabolic
showing a
processes.
protein
Catabolic
core,
processes
with a
are
concentration
things that
being
break
higher on
things
one side of
down.
the cell,
As soon as I
lower
get
on
done
the
with this
other
lecture, I'm
side
going
of
down to the
cell.
gym.
Facilitated
Work out on the
diffusion
treadmill in order
is
to
simply
hopefully
diffusion
have some
where a protein
catabolism
core assists the
occur,
movement
and
of
my
molecules
goal
through
is
the
to,
cell
by
membrane.
exercising, to lose some weight.
Another type
Other
of
people will go to the
transport
gym
is
and build
osmosis.
muscles,
Osmosis
or
is the diffusion
lift weights
of
with the
water
help of
across
building
a
muscles.
semipermeable membrane.
That is an anabolic
Again, that idea of
chemical
water moving
reaction.
across a membrane.
Again,
you need to think a
more
little bit about diffusion.
reactions or
Diffusion
more
is moving
pictures
up
of
from
these
high
things
concentration to low concentration.
reactions
And when we
together.
look at this thing
Again,
here,
you need to
here's
tie
a
the words
solution on two
catabolic,
sides of the cell.
exergonic, and oxidation together.
On one side,
Anabolic,
there's
endergonic, and reduction together.
a solvent, which is the water. You can see the water molecules.
And finally, your
And there's
metabolism is the
more
sum
solute
of
on
both
this
your
side, and on
catabolic
this side,
and
water
your
tends to
anabolic
move
processes.
towards where there's a higher concentration
Okay,
of solute.
so
a higher
this is the first place
concentration
where we're going
of
to start
solid
talking a little
particles
bit about enzymes.
until
We know enzymes
equilibrium
are
is reached.
necessary for all the chemical
Okay, so
reactions.
can describe cells that
enzymes
have
are
different
proteins.
concentrations of solution.
All of our biochemical reactions are going to
Something that
require
has lower
enzymes,
concentration of solute,
and so
we say is hypertonic.
enzyme, we need to know
Something
the
that has
second
higher
bullet point
concentrations
is a
of solute,
biological catalyst. It
hypertonic.
speeds up or pushes forward a reaction.
More energy.
Again,
we think about
there are
hyper
going to
people.
be
Hyper
proteins
means
that have a
more
three-dimensional
dissolved
shape
solute.
somewhere
And isotonic means
in
that
that
there's
shape is an
equilibrium.
active site.
So
The
water
active
will
site is a
always
place
move from
where
a hypo to a
the
hypertonic
substrate
solution.
or the
A couple ways to think about that.
where the
Water
substrate
always
can bind,
moves towards where there
and
is more salt
it's
or more
absolutely necessary for
solute.
enzymes to
So
form
if
an
you
enzyme
have,
substrate complex, for enzymes to bind to their
an
substrate.
animal cell, if you have like a
before
water cell,
product can be
and you put it
produced.
into
When an enzyme binds to a
hypotonic
substrate,
solution,
okay,
what happens
the
is
substrate can be
the cell is hypertonic,
go through a
water
chemical
moves
reaction,
in,
and products
it'll
will
cause the cell to lyse.
released.
If you have a
What
isotonic
happens is
solution,
the
water will move in
enzyme
and water will
lowers
move
the amount
out.
of energy
And if you
necessary
have a
for that
hypertonic, that means
energy,
there's
that reaction
more
gets
solute.
started. So normal
On the
amount of
outside,
energy
again,
to start a
water
reaction
always
is called
moves towards where
activation
there's more
energy.
solute, and the
If
cell
the
will
activation
shrivel.
energy is
You
usually
can see
too
these
high for
examples
most
in animals
reactions
and
to
plant
occur
cells
spontaneously,
of water moving
enzymes
from
simply lower the amount of
hypotonic to
energy to get the
hypertonic
reaction
solutions.
started,
You
which
need
is
to
usually
memorize
enough
that.
to
Water will move
cause energy to
from
be
hypo
released.
to hypertonic solutions.
There's a model that we now use
Now we've
for
talked so far about
how
passive
enzymes
transport
function,
doesn't
and we
require
say
energy
that
from the cell.
an enzyme we
We're going to go on
know
and talk
is
about
a
active
protein.
transport.
Proteins
Active
have
transport generally moves things
hydrophobic, hydrophilic
against a
sites, and
concentration
those different
gradient.
sites
Again, it
can
moves it from
attract
low
a
to high
particular substrate.
concentration.
Any substrate
It
can
requires
form
the use of
into a
cellular energy,
particular
or
site.
ATP.
When the substrate fits into the site, it can alter
This
the
is going to
bonding in
require
that protein
movement through proteins,
just enough to cause stress
or again,
on the substrate.
endocytosis is described
And that
as
stress
a
on the substrate
active transport process.
changes
Now, there's
the
some other
reaction
processes
between
if you
enzyme
take
and
some higher level
substrate
biology,
in a way
you learn
that
about
promotes the
symport
production
as
of
an
a
active
particular
transport
product.
process. But that again requires
Okay,
the building
so
of a gradient,
couple
which is an
key
ATP
things: enzymes are not consumed or
ATP
altered during
requiring
chemical
process.
reaction. The enzymes don't change.
Here you can see
that
again
means that
a
enzymes
solute
can be
moving its way
low
into a
at
transport protein
effective
and
at very low
ATP
concentration,
activating
have
the
one
transport
enzyme
and movement
catalyze
of that solute
many chemical
from an area of
reactions.
low
Again,
to
here you
high
can see it.
concentration.
The
This is
enzyme
sort of
sucrase, a good
which,
description
with
of
a
the
standard
substrate
active
sucrose.
transport
Sucrose
protein.
fits into the enzyme.
Here's a picture of
Again,
endocytosis.
when the enzyme
Again,
substrate
endocytosis,
complex
the cell membrane
fits in, it
folds
causes
inwards,
the production
surrounds something,
of,
forming
or
an
causes the break
endosome
of
or vesicle,
the sucrose,
and
which
the
is a
vesicle makes its
disaccharide,
way into the
two
cell.
monosaccharides. Products are released, and the
Again,
enzyme is ready to do the whole
exocytosis
thing all
is
over.
similar to endocytosis, only going in the
There's a
opposite
relationship
direction.
between
In this
temperature
case,
and
a
enzyme
vesicle
activity.
is fusing with
And
the cell
you can see that we
membrane
have the
and dumping
Siamese
its
cat
contents
here.
outside the
Siamese
cell.
cat has some really
Again,
interesting
all these
enzymes
processes require ATP.
that can produce pigment only when they're
So
cool.
far we've talked about
so you can
passive
see
and
Siamese
active
cats
transport.
actually
Passive
have the
transport
ability to
included
produce
diffusion,
pigment
osmosis,
throughout their
and
entire
facilitated
coat.
diffusion.
But you only see pigment
Active transport
production
included
occurring
protein
in the areas
pumps,
of the body that
and
are
also
relatively
endo and
cool. In theory,
exocytosis.
you could tape an ice
And
pack
again,
onto
these
this
are
cat,
models where the
and you
cell
would end
uses
up
energy
with
to move
dark
things
pigmentation
from an area of
where
low
the ice pack
concentration
is
to an area of high
placed.
concentration.
So it's
Passive
a really
transport
neat
is
picture
always
of
from
enzyme
high to
activity.
low concentration.
Every enzyme has an optimum temperature.
Make sure you
Every
review this. We'll
enzyme
go on
has
to
an
the second
optimum
half
pH,
of
and every
chapter
enzyme has
5.
an optimum of other conditions of the solution that promote its activity. Okay, here's a neat picture of the relationship between pH and enzyme activity. This enzyme here that's described by the black line is actually pepsin, which is an enzyme that's active only in your stomach. What happens is this will help begin the breakdown of polypeptides or protein molecules. Your stomach has a pH somewhere around 1 or 2, so that will promote the activity of pepsin. When the food leaves your stomach and goes into your intestine, buffers will raise the pH somewhere around 6 or 7, and at that point this enzyme pepsin can no longer work. On the other hand, most of the other enzymes in your body probably work best in a pH somewhere between 7 and 8. Again, every enzyme has its own optimum pH. We can go on and talk a little bit about enzymes. Again, most chemical reactions are somewhat complex and they take multiple steps. There isn't a single step that causes them to occur, but they're a multiple step process. So we often find that the product of one enzyme, enzyme 1, will be the substrate for a next enzyme, which is the substrate for a next enzyme. And ultimately, what can happen is as a final product builds up, it can actually act as an inhibitor and stop the activity of enzyme 1. Again, what's neat then is you'll have no more production of the product. Product will be possibly used up, and once it's used up, the product will stop binding to the initial enzyme and allow this reaction to occur all over again. This is called feedback inhibition, or negative feedback, where the production of a lot of product stops the production of more product. Again, metabolic processes occur in multiple steps, and this is a beautiful diagram of why. This is showing you hydrogen and oxygen used. If you add a spark to hydrogen and oxygen, you can actually produce water. Of course, you'll also have an explosion. In the cells, hydrogen and oxygen are added together for the production of water during cellular respiration, but it's a multiple-step process, and by releasing a little bit of energy at a time, there's nothing explosive going on, and again, some of the energy can be trapped to do other things. So the next question is, enzymes allow all the chemical reactions to occur. How do we regulate those enzymes? And the answer is, it depends. One option would be denaturing the enzymes, but the problem with using something like heat or changing pH to denature enzymes to control them would be that they would denature all enzymes, and they could really affect the overall function of a cell. So cells, rather than denaturing proteins when they want that to slow them down, will actually use what we call inhibitors or activators. I'll often talk, if you ever have a chance to watch children are a beautiful way to think about enzyme inhibitors and enzyme activators. A child will be super active all day long, and you put them to sleep. It's almost like their whole metabolism shuts down. You can imagine chemical inhibitors just saying, slow down, we don't need to produce any more energy, and storing up the energy that they'll be using for the next day. Again, our enzymes have a variety of mechanisms of regulation. For the most part, we use chemical inhibitors and activators to control specific enzymes rather than controlling enzymes with temperature or pH, which would not be enzyme specific. And again, here's a nice picture of feedback inhibition. The nice thing about this is the feedback is reversible. So here's a neat picture of what we call an allosteric inhibitor. An allosteric inhibitor, allosteric means other shape, is something that affects the enzyme by changing its shape. Again, normally in this case the substrate would fit into the active site. An allosteric inhibitor will fit into someplace other than the active site and change the shape of the active site, and that can inhibit the way the enzyme functions. Now this one here is a competitive inhibitor. In this case we have another molecule blocking the active site. Okay, again, this is a competitive inhibition process, this is a non-competitive inhibition process, and both of these actions can inhibit the activity of an enzyme. We can also have allosteric activators. In that case, allosteric activator can fit into a binding site and promote the activity of an enzyme. Again, without that activator being present, the enzyme may not be working. So let's go on real quickly and talk about coenzymes and coactivators. These are very often things that help an enzyme to function and without actually being part of the enzyme. Again, sometimes they donate electrons, sometimes they carry molecules, sometimes they bind the enzyme to improve the shape of the enzyme. Now, there's two types of molecules that can help enzymes. One of them is called coenzymes. Coenzymes are large organic molecules, and most of your vitamins are actually coenzymes. They help enzymes to work better. We can also have cofactors. Cofactors are small inorganic molecules. These are mostly the minerals. So you may have been told you should take vitamin C to promote your immune system. And that makes sense because vitamin C is one of those molecules that helps the immune system to carry out its functions. Likewise, you may have heard that zinc, which is a cofactor, can help again improve the activity of your immune system. Now, a quick question is, do you need to take vitamins and minerals? And the answer is no, if you have a completely balanced diet. That's a big if. Each person needs to make a decision for themselves if it's appropriate to take vitamins and minerals. That's it for the activities or the understanding the basics of cellular energetics. We'll move on in chapter 6 to talk about cellular respiration, which is one of the most important energetic processes.